Hello, good morning, good evening, everybody. I am Vaidya Asavari Manavikar. I am from Minnesota, and I would love to welcome you all for GCAR, Global Council for Ayurveda Research, our webinar research series. So, Global Council for Ayurveda Research is an international non-profit organization based out of USA. Our mission is to encourage, educate, and facilitate basic, clinical, and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence science which is based out of globally. And you can find us, like you can find more information about us at www.ayurvedaresearchusa.org. Uh, Please consider to joining us as a member, which will bring you access to all our resources. So there are lots of projects we do and everything is on the website. So you can definitely have access to all of those. No nonprofit can operate and succeed without the volunteers. So we would love, if you are interested, definitely reach out to us and let us know about your skills, your interest areas, and we can place you accordingly. Once again, our website is www.ayurvedaresearchusa.org. Now, let me share and introduce our eminent speaker today, Dr. Ashwini Godbole. Dr.
Yes.
Ashwini Godbole is an associate professor at the Center for Ayurveda Biology and Holistic Nutrition, TDU. She has made significant contributions in Rasayana therapy research, with a special focus on aging and neurodegeneration, which is a very, very, very important topic, as you know. Her ongoing work explores the impact of Ayurvedic drugs on lifespan and aging related health through both community based clinical research and experimental models. She has many scholarly publications under her belt. Today, she will be speaking about Medhyrasayana for healthy aging and cognitive wellness. Let me
Namaste, everybody, and thank you. Thanks, Asavari. Thanks for the introduction and the invitation, both. And I can see some of my friends have also joined here, so happy to present my whatever research efforts we are doing at TDU. TDU is also an interesting place, so I would like to take this opportunity to invite everybody to visit TDU, visit TDU website, and see what research we are doing, and we are always eager to collaborate and work together with people. So with that introduction, I'd like to share my slides, the presentation. I hope everybody can see it. uh, so I should tell you, take a little bit of time to tell you all that I joined. I'm not educated in field of Ayurveda. I'm not by training. I'm not a Vaidya. I'm a biologist. I did my PhD in cell biology and biophysics. So it was a totally different field. But I always like to do something which matters to people. And after spending my time in fundamental research, I thought I want to do something which is close to close to India, and also means something which is born in India, which is very much Indian. Also something close to plants, because I'm a botany graduate. So both together, and interest in cultural and traditional things all put together, and I found TDU. So then I started working in this field. When I started working, I was in a project which was working on Rasayana. While I was very fascinated by that concept itself, and I have a slide on that concept from Ayurveda called Rasayana. And then I slowly matured, and we chose Medhyrasayana as a topic for research, where Medhya is something which is very good for nervous system, mainly brain. So we thought this is a very interesting topic, and as a biologist, I felt that this I want to to research more into this and understand how things can be changed at various levels, because as you understand, the nervous system has reflections at the behavioral level, at the cellular level, at the molecular level, and lot of connections with the whole body as such, health of the whole body. So that's why, and psychology and mental health. So I thought this is some area where we can do a lot, if and if we can show that Ayurveda has these things to offer to the world, and then we can, if we can show it with through systematic research. So this is an effort for that. Please do comment, suggest, or collaborate. Let's discuss more at the end today, or even in future whenever you feel like. Please do connect. So, coming to the today's topic, healthy brain and mind. None of us will deny that both these things are very important. Both these things, first of all, are very different. Healthy brain and healthy mind are two different things, and both are very important. And Ayurveda gives importance to both these things. Mostly, concept of in Ayurveda, if you see, it keeps on talking. Ayurveda says about manas and not about brain as such. Brain, of course, there is a description of brain in Ayurveda, but manas is something which it gives a lot of importance to. So I thought there is a lot to offer from Ayurveda, and my colleagues from TDU also, we get to collaborate with a lot of Ayurveda experts, and then that's how we come to this thing. Another fascinating thing which I about Ayurveda is that what it gives is a lot of importance to health, maintaining the health and wellness, and then illness is comes later. The more importance is given on wellness, and so that we don't fall sick, there is no problem. So if we maintain our health, if we are well, we will unlikely to get any disease or disorder. So that's why swasthasya swastha rakshanam is something which is another attractive thing of Ayurveda. And when it comes to nervous system, we know that the health means enhanced structural features, means enhanced physical strength of the nervous system, the development, good development of nervous system, maintenance of the nervous system structure, and regeneration. So we know that stem cell field is very popular now, and we know that there is limited but good amount of regeneration even in nervous system. Enhanced function, even if the structure is good, it has to be reflected in function, and enhanced function is another parameter for health and maintenance of. So having good function, having good structure, and maintaining it will automatically keep the disease low, and the chances of getting disease will be automatically low if the health is more and enhanced. So that's the degenerative, the functional problems and the degenerative diseases both. So when we say nerve, good health of nervous system, it has to be good sensory functions, good motor functions, good cognition, which is very important, and tranquility and happiness. So if all these things are good, most of the time this can be achieved, and this is a very important factor of mind, tranquility and happiness. If when we talk about, even if we don't like, but if we talk about diseases of or disorders of nervous system, then there are functional problems like stroke and epilepsy, and then there are age related degenerative problems which are like Parkinson's disease and Alzheimer's disease. And we have good amount of our research uh, focuses on these aspects which are age related and not so much on the functional disorders. So when we talk about cognition, we know that there are different aspects of cognition. Cognition is memory, attention, and concentration. Three aspects of cognition, and in different stage of life, there are different things which are good. We are good in some things, and we are not so good in some other things. So it's not that only later ages have problems. We see that children are very good, they grasp very quickly, their memory is good, but attention is often a problem. In youth, you have learnt everything, now you have the information, now is the time to implement. And that's when too many things, managing things, then stress, sleep problems, and associated memory problems can come in, and we can manage it well if we follow some advice from Ayurveda. at later ages and the senior ages, we see that there is a lot of wisdom. It's not just enough to have knowledge. It should be distilled, and it is converted into wisdom because of the experience also, which is attached, which is along with the knowledge. And then comes the problem of memory, losing memory or reduced memory, sensory and motor deficits. And all these cases, be it in childhood, be it in young age or middle age, or be it in senior ages, we can manage these things, which can be otherwise a challenge. As we can say, we cannot, it's not that all the things decline with age. There are certain things which remain stable, like numeric ability and verbal ability remain stable, but something which declines very much is the fluid intelligence. The quickness, the fluid intelligence are something which decline. The crystallized intelligence remain the same. These are certain things which we consider while designing our research, and the way we do the research, where we do the tests, assessments. So, what we want to achieve finally is that we want to check if there are solutions from Ayurveda which can help us in doing achieving successful aging. Because, as you can see, cognitive aging can happen faster than the physical aging. That's why we need to make sure that the cognitive aging does not happen, or it's very slow. It's reduced, the speed is reduced, and you can that will be successful aging. So that's the aim of the study, that we want to see if Ayurveda have solutions, and if we can do show them scientifically, if we can do the show the science behind it, then that would be good. So this is what we are trying to do. As everybody knows, but I'll just sake of continuity, I will say that the modern biomedicine, both the practice and the research has to offer us the details. What happens at the molecular level, what happens at the cellular level, how things change, whether we have this area, we can do a lot of analysis by using a lot of instruments, and say that whether the structures are maintained, or so many other things. Those parameters are objective. That means whether I do the reading or somebody else does the reading, there is not much of a difference. So because many of those are machine based, so those are kind of objective parameters. Uniform, so the treatment is uniform, and that's why it is scalable, and the availability is there. So because things are uniform, there is availability is also not a big challenge. So, and in traditional medicine, we have holistic approach. It is very personalized. So, and the emphasis is given on health and wellness. So these are the positive points, and of course, both these systems have the flip sides, which is lack. The modern medicines doesn't look at the system, doesn't treat the individual as an individual, but and take the systems approach. It also can lead to, especially with nervous system, it can also lead has many modern medicines have a lot of side effects, like medicines for Parkinson's, medicines for Alzheimer's. They have their own side effects and limited efficacy with respect to chronic and lifestyle health problems. So there are no quick fixes for these kind of things. So there is a problem, and lack of wellness and prevention. Lack means there are very few wellness and prevention solutions. But on the other hand, the traditional medicine, there is lack of documentation, as we understand it scientifically now. There is documentation, there is literature, but the documentation of cases, documentation of results, the what has happened before and after the treatment, and so on, is not in the way the modern scientific world will like to have. So that's one flip side. There are complicated treatment protocols, and because it is very skilled or resource dependent treatment, like the herbs are required or some things have to be prepared in a particular way. That's why the resource availability or scalability is limited. That's why what we thought is that we can look at both. We can have a transdisciplinary approach where we can take good from both the sides, and then mix it to make a transdisciplinary protocol or transdisciplinary approach towards the nervous system health. what is accepted in even in modern practice now, and more so emphasized in Ayurveda, is that such kind of lifestyle changes, what you eat, how you eat, how you what do you do over the day, whether you do have physical activity, no physical activity, and so on, and as well as what is the supportive medicine or supportive things which you are taking, which might change with the season, which might change with age, and so which might change with the area, geographical area you are staying. So that also is important. So as I said, we try and do transdisciplinary research where we use concepts and practice from Shastra and facts and findings from science and the tools, and we mix these two, utilize these two in as optimum way as we can think of, or we can we are doing, which is available here, and we then do the research which will give out some integrative kind of information where we will have we can apply it in current condition, contemporary health needs, taking in information from the traditional medicine. So we are working on three different areas, that is utilizing three different things. One is the foods, the nutraceutical herbs, and everything to do with Medhyrasayana, the something which is good for my health, brain, and nervous system. So these are the health foods which are highly recommended in Ayurveda. Ghee is very highly recommended. Almond is very highly recommended. Walnuts, and of course there are recommendations like fish and milk also. Then there are nutraceutical herbs which are actually called as Medhyrasayana, like the herbs, Medhyrasayana herbs, and we have those in Ayurveda. Uh, and we also have other Medhya Dravyas which are supportive medicines or supportive herbs, which are also used largely in practice for effect on memory, effect on stress, or something to do with the nervous system. Our hypothesis is, when it comes to food and formulations, and from the cell biology perspective or from the molecular biology perspective, the neurobiology perspective, we know that there are changes in the nervous system which are depicted as molecular damage. So all of you must, most of you would be knowing that there is deposition of proteins in the brain as the brain ages, and if the deposition is of a dangerous or toxic protein, something like A beta in Alzheimer's or Parkinson's, then there is a damage to the tissue, damage to the cells. Then there can be energy dysregulation. So that's at the functional level. So it's not so much visible at the structural level, but because the mitochondria don't function properly, the energy regulation is not okay, oxidative stress is there. That's why there can be that leads to oxidative stress, and then that can be a problem. Immune disorders, brain also has immune cells called as glia and microglia, and those can function, over function or under function, and that can lead to a problem. And all these things, if are not under control, can lead to neurodegeneration, which is like Alzheimer's, dementia, Alzheimer's, Parkinson's, Huntington's, similar kind of thing. And if we adapt, or if we build resilience in the system, which can be called as adaptation, to tolerate the stress, to adjust according to the condition, and to overcome the stress and stress related problems, then the system can function properly. And in Ayurveda, as I understand Ayurveda, and this is our hypothesis, and we are trying to test those things out in the experiments and as well as in the clinical study, we see that the spices are there, or the something which is pungent, something which is sharp, normally cleans out the system, and that is called as Shrota Shodhana in Ayurveda. Shrotas are the channels or micro or macro channels, and those are cleansed. The system is kept clean, and that is what is most likely. If you take something which is like spices, that might be the job it might be doing, and that we can check by in the experiments which we are doing. Agni Vyapara, that is maintaining the metabolism, maintaining the energy regulation, and that can be that is Agni Vyapara, and Ayurveda suggests lot of Agni Vardhak dravyas for that, and that is applicable also for the nervous system. To keep the nervous system healthy, to build the resilience, to reduce the aging, For all that, Ayurveda suggests Poshana Dravyas, and many of those which we use every day in food, as I said, which is ghee and nuts and milk and so on. And they can be high in antioxidants, and what is very very intriguing and very no, very unique in Ayurveda, and is the application of fats, use of fats, like oil and ghee and so on as base, especially when it comes to nervous system. So we are doing, as I said, we are doing three things. We look at Ayurveda very carefully, the theory of Ayurveda, the concepts from Ayurveda, and we have in our team, you will see at the end that we have Ayurveda doctors, we have neuroscientists, and we have MRI scientists, and so on. So we do all cognitive neuroscientists. So we all together do this research. So we look at the theory and the concepts of Ayurveda. We do the clinical research and we do experimental research. We use we use various systems for doing that, and also of course we use a lot of data from clinics or community-based clinical studies. So, as I said, just to reiterate, we use concepts when it comes to media, and I will talk more about it as I move forward. So, there is a concept called pragya, that is cognition. That has a lot of parallels with cognition. That has we can connect, we can draw a lot of parallels from concept of pragya to cognition, and we have looked at it, and we see that there are three aspects of pragya: buddhi, medha, and smriti. And buddhi is something which is working at the sense organ level, or we can say that, and that will filter out the noise from signal, meaning even if you're going on the road and if you see 1,000 cars passing, you don't remember everything. You will only see the whatever you want to see, maybe the red signal, and when one which turns green is what you are aim at, and you know what has to be seen and what has to be ignored. Then medha is the information which it gets filtered, and some information gets converted into memory, which is smriti, and some is kept, like there is a filtering what to convert into memory and what not, and what kind of memory also. And Medha is at a very critical stage where Smriti, whatever we remembers, also has to come back to behavior, and that function also goes through Medha. And that's why I think Ayurveda gives a lot of importance to this entity called Medha, and that's why the name Medhya Rasayana. So, because it is right in the center, where on two sides buddhi and smriti, and on the other third side behavior. So for all this, medha is that very crucial thing, crucial place, and medhyarasana is maybe what supports medha in general. As I've already said that about rasayana is one concept, and we have three modes of action postulated in ayurveda about it. So what we do, we have selected certain herbs and formulations, looking at how they are used in practice, also talking to the experts, and then selected those. Brahmi Grita is something is of which we are looking at very seriously. Brahmi, when Ayurveda says Brahmi, then there is Mandukapani also along with it, and Mandukapani is supposed to be very good. Rasayana, as many people know that these are also colloquially both are called as Brahmi. This is used, Mandukapani is used in food, it is used in chutney, it is used in curries, and it's very good Methi Rasayana, supposed to be very good Methi Rasayana, and we have some data for that. We have Brahmi Grita data for the clinical level as well as the experimental level. Another third thing which we are using, working largely on, is Kshiravalla, which is for motor health. So these two on the left hand side, as you see, Brahmi and Mandukapani are for cognitive health, Kshiravalla is for motor health, and all three of these are used for in age related decline, and that's the main interest of the group currently. Age related decline in nervous system function. So that's why we have chosen these three, and we are working majorly on these three. There are some other small projects which we are doing on ashwagandha, mucuna pruriens, that is kapikachu, shankapushpi, and nishamlaki. Vacha, for that matter, is again given, Acorus calamus is given as neurotonic, again used for nervous system disorders, mainly stroke and epilepsy. But you should see that maybe the Ayurvedic doctors can comment on this. But what we have seen is that it is mainly used for functional disorders and not for degenerative disorders, and that's why we use that as a biologist, as a scientist, we call it as negative control. So something which might not, mostly will not work in degenerative disorder, will not stop degeneration, but will help enhancing the function where the degeneration is not a problem. All other three on the top are used for degenerative disorders. So age is one of the things, factors which leads to degeneration. So these are the kind of logic which we have used while doing this research. So community-based clinical studies, this emphasis on Brahmi Grita, and the hypothesis is that Brahmi, especially in Grita form, can mitigate age-related cognitive decline. So that is our research hypothesis. We are saying that Brahmi Grita, which is highly recommended in Ayurveda, practiced in Ayurveda, if you go and if you the Ayurvedic doctors will agree with me, wherever there is any cognitive problem, anything to do with memory, the first choice is Brahmi Krita, or something which has Brahmi, maybe Saraswata Rishta or something. But many times, if it is given for age related cognitive decline, it is the Brahmi Krita which is the first choice. What do we do? We do a lot of, as I said, we do transdisciplinary research, and we do a lot of we have neurocognitive scientists with us, who are who are who do cognitive assessments, which are paper pencil based assessments, where we can check memory, attention, concentration, speed of action, speed of doing things, speed of how well they understand, how well they remember, short term memory, long term memory, and so on. We do check, we also do MRI, which is, we also have functional MRI aspect in it. We do stress, anxiety, depression, sleep scales. So all this has a connection: sleep depression, anxiety, and stress with cognition is what we feel. We do MRI, as I said, as an objective parameter, and we understand a lot from which area, from functional area, we understand which area of the brain is more active, how the function changes with age, and so on and so forth. Then we do before and after blood test. So this much from the modern neuroscience point of view, the three things and blood test. Then we also look at Prakriti. We do detailed Prakriti analysis. We do Agni analysis, and we try and see that, try and correlate it with person's cognition, and which formulation has helped or not helped in which so a lot of parallels can be drawn, but a lot of correlations can be drawn if we have detailed Prakriti analysis and the person's thing. So we, at the end of the thing, we tell the participants that their Prakriti is so and so. They, some things are, these are their strong points. These are some of the weaknesses. How they can deal with it, and so on and so forth. And then we also tell them about their cognition, and how was it before Brahmi, how was it after Brahmi, and what are the take homes from that research. So we do give advice to the participants, and of course all this is done. So we have, as I said, we have all these. We have four formulations Brahmi ghee as a complete traditional Brahmi ghee formulation. Which has, of course, has base as plain ghee. So we have also a group which is plain ghee, because Ayurveda says that ghee itself is good. So, but we want to see, when it comes to age related cognitive changes, whether ghee is enough, or Brahmi ghee is better, or how does it work. Then we have a already commercialized formulation called Bacomine, which is a Brahmi extract, very high in bacosides, which are supposed to be one of the active components of Brahmi, that is Bacopulmonary, and we have placebo group. So we have these, and the study period is for 3 months. And as you can see, in nutshell, we are still collecting some data, but in nutshell, if you have to compare Brahmi extract and Brahmi Grita, and this was very surprising for us also, you can see Brahmi extract has some effect in younger population, which is not, we have this, by the way, this study is assessor blinded study. So the person who does assessment does not know what the participant has got, whether Brahmi extract or Brahmi Krita. Brahmi extract seem to be working in Brahmi in the younger population, which is 25 to 55. Brahmi Krita does not seem to have much effect, and we should remember that this population already has good cognition, normal cognition, and there are not much changes. When the cognition is little bit on getting lower, declining in 55 to 85 for age group, surprisingly, Brahmi Gupta just works wonders. It just works on all types of memory in people who are maybe at higher age, above 55 years. So this was very pleasing. We have got almost 200, 200 participants in both the groups, and taking different formulations. So here we have put above, over and above the control, the placebo control, Brahmi extract shows effect in younger population, especially sleep and quality of sleep. It was very nice to see, because as I should tell you, that in our community, in urban Indian community, we saw that younger population has more sleep problems than the older population. Older population sleeps well. Our elderly people who are above 55 are they sleep well. I think it's the digital media and work and everything put together. The sleep quality is bad in younger population, younger group, and that actually Brahmi extract helps. So that is something very we feel is a good result, and Brahmi Krita helped in the older population for all types of memories and other parameters also. From the MRI, we saw from MRI studies we saw that as the age goes up, there is change in certain neurometabolites, which are the metabolites in our brain, the chemicals, the neurochemicals in our brain. There are changes in that, and one of that is choline, and other one is myoinositol. So you see that choline increases with age, and myoinositol also increases with age, and it was very nice to see that just 3 months treatment with Brahmikrita actually lowered the choline levels, which is kind of anti-aging effect. We feel is an anti-aging effect of Brahmagupta. So this is what we have got, and it was before and after Brahmikrita treatment is what we compare the choline levels. We did not see that kind of effect in myoinositol, but we did see in choline, and we are doing various other markers and areas of brain which we are looking at whether Brahmi has an effect at the structure or the functional level, Brahmi Krita and Brahmi Extra. When we look at, so this much we can get from the clinical study, that Brahmi Krita is working on memory, Brahmi Krita is working at the brain level when it comes to neurometabolites and so on and so forth. But then when we want to see whether it is anti-aging, what level is it working, which molecule is it reducing, what kind of cellular changes it is bringing, so all that cannot be done in the clinical setup. It's difficult to do with human beings, ethically also and technically also. Technology doesn't allow us to do that. So we are doing experimental research, which we call as mode of action research, where we are looking at something similar to cognitive functions, anti-aging effect, and sensory motor functions. Why anti-aging here? Because there is lot of data available, and Ayurveda also says that age and nervous system decline, increase in age and nervous system decline goes hand in hand. If you reduce aging, nervous system also remains healthy. If nervous system, something which offers health to nervous system, the corollary will be something which is offer health to nervous system should be anti-aging. Or so that's what we are trying to check. And as you can see, some people have already, this is not our data, but just for sake of showing it, making a point, I am showing this. Some people have already shown that Brahmi as extract has effect on brain. You can all easily make out that this is a neuronal cell from a rat brain, and you can see cell in A and cell in D is different, isn't it? So if the cell in A, I'm sure it's very obvious to everybody. What can we see? There are less of processes, there are less of these branches, which are called as dendrites, and These That means there is less of dendritic corporation, and just 80 mix per kg of Brahmi extract has made this change. The neurons have much more branching, and as you know, as we are human beings, we are more socially connected, more healthy we remain. Similar goes for neurons. More connected are the neurons, more is the branching, more connected those neurons are, neuronal cells, healthier they remain. It's less likely that the neuron will get damaged. That means there is more of resilience if there are more synapses or more connections, which is indicated that there are more branches. That means there are more connections, and that is what is shown in rats here by another group. And what we do, we use majorly the C elegans as as a model. It's a small nemator, and which has only 1,000 cells. Out of that, 309 are neurons. So one third of the body is neurons. It is transparent, so we can mark the neuron and see. The lifespan is 21 days, so we can finish our studies. We can feed these tiny little worms, the larvae, and we can see only Brahmikrita only in that stage, middle age or young age, and see whether it has effect on the older age. Because Ayurveda also says if you take care of yourself in early or the middle age, you are less likely to have problems in the later age. So all these things which is difficult to do with human beings, we can do with these such kind of model organisms. Again, we have models for learning and memory, we have models for Alzheimer's and Parkinson's like disease, and we can look at aging, of course. I'll show you a little bit of data here. Stay with me because we can see something interesting. As I said, the worm normally lives for 21 days, but if you feed Brahmigrita to it, it lives 1.5 times more than 1.5 times, and it is much healthier. It moves better. It looks younger. If you see worms under the microscope, it looks much younger than its other counterpart, the control group. And ghee also does the job pretty much okay, but not as good as Brahmi. So Brahmi Krita has added advantage over the plain ghee. And Brahmi Swarasa, that is juice, and Brahmi oil. Brahmi Grita in this case stands much better, even with as compared to the Brahmi Swarasa and Brahmi oil. So there is something in Brahmi ghee which offers it anti-aging and neuroprotective, both the properties. So that's what we have seen in our research. We can also test memory of the worms. So they like certain things and they dislike certain things, and we can teach them that this thing means food, or this thing means shock, and then they will get repelled. And as I can show you here, I'm not going in details of the experimental procedure here, but you can see that the control worms and both the Brahmi fed worms learn. They learn that something is good or bad for them. But after 1 hour, the control worm, normal food fed worm, forgets it. But the Brahmi fed worm remembers still that this is not good for me. So that kind of memory, the association is lost in the control group. That means the memory is gone, but Brahmi fed worms do remember still. Half of the worms still remember that this is something is good or not good for us. So that's the kind of experiments we can do, and that gives us advantage. As I told you, we have models for Alzheimer's, and the protein which causes Alzheimer's in human beings is expressed in the body wall muscle of the worm. And you can see that when the worm gets Alzheimer's, kind of, it stops moving, it gets paralyzed. And when the worm is healthy, it moves along the whole plate, and you can see the tracks. And as you can see, little bit of Brahmi Swarasa actually completely stops this kind of paralysis, and it's very effective. So that's how we have seen the effect of Brahmi till now. Similarly, people have shown ashwagandha. We have also done studies with ashwagandha, but I just want to show that similar model, a rat but mice model of Alzheimer's. You can see when fed with ashwagandha, you can see the blobs here. These things, the brown dots are aggregates of the protein, which actually leads to nervous system degeneration. Degeneration means death of the neurons. So you can see, but such blobs are not there when the mice was fed with ashwagandha. And similarly for hippocampus, which is a working memory center for that. So similar effect, that means that we can see, look at the level of organism and look at the level of human beings. Both is applicable. Why is it, why does it make sense? Because we know from the science, the modern science, that the genes which are responsible for nervous system function, genes which are responsible for aging, are all same. Be it the worm, the C. elegans, Caenorhabditis elegans, be it mice, be it monkeys, and then human beings. The aging cells are very, aging genes are very well conserved. Aging pathways are conserved. Similarly, nervous system function pathways are conserved. That's why we can use these animals and these organisms and these systems as proxy for looking at something as clinical effect. Yes, so similarly, one very other interesting data from Ashwagandha paper is that these are the blobs of the protein, which are bad proteins, and these are the soldier cells. And when ashwagandha is fed, the soldier cells are more active, the immune cells of the brain are more active, and they clean off the garbage protein very quickly. So that's the kind of data which we get from the science, the neuroscience research, which is on these kind of Medhyrasayana for herbs. So we are so the hypothesis here, or what the experiment have shown, is that ashwagandha does not directly act on brain, and that kind of information also comes from the experimental research, because we have the markers which we can check in the animals. It does not directly act on the brain, it acts on liver, and then it makes a protein which goes and sits at the blood brain barrier and pushes away all the garbage protein out from the brain and is excreted out. So that's how it works. So this mode of action information, I feel and we feel, and you can comment and you all can tell us, builds up a confidence in or understanding, deeper understanding in what must be happening when you prescribe ashwagandha, brahmi, or bala, or any herb or any formulation to patients or the people. Ask them, recommend it for people for good health and wellness. So if we know that it works at such and such level, it makes that difference. It is always very much more convincing, and I feel that it is more important for wellness, because if person doesn't have any problem, it's unlikely that person will use do some lifestyle changes, add some of these herbs in the routine, and so on. But if we show that these are have anti-aging effects, and if we use them in the middle or the young age, it is going to have an effect at the older age, or even if you use it at senior elderly, for elderly people, then also it will be a beneficial. It will build resilience, it will build health and it have wellness. So that's how we want to that's what it will be. These kind of this kind of research will be more useful, both at the disease and mainly for the wellness kind of interventions. So, what I have shown now are some pieces from the jigsaw puzzle, and as you know, that to make this puzzle more and more complete, to get the complete picture, many such work have to go on, many such efforts have to be there, and that's when we will be able to get almost the complete picture. Rest of the pieces can be imagined and put together. So thanks a lot once again for this opportunity, and I should thank our funders and the institute. TDU is a very fantastic place where we have people from many different areas. As the name suggests, it's really transdisciplinary. My uh, clinical team is very good. The support which we get from the other places like IISc, and it's good. We are getting funding from Pratiksha Trust, which is Chris Gopal Krishnan, Infosys founder Chris Gopal Krishnan, his family trust. We get funding from RIST, which is Rural India Support Trust, and we get all the support from our hospital, which is IIM, and all other colleagues. Our team is very good. We have people from clinic, Ayurveda, cognitive neuroscience and people working in the lab. So thank you and happy to connect, happy to take questions and comments, suggestions, anything. Yeah.
Thank you so much. This was really very, very in-depth, very informative.
Thank you. Yeah.
Amazing, amazing presentation. Lot of work, research you have done for so many years.
Yeah. Big team.
I mean, Ayurveda is so vast.
Teamwork.
There is
Yeah.
so much knowledge and wisdom, but it needs to be proved. There needs,
Yes.
some data in today's life.
Yes. Yes. So I would say that we just need to show the science behind Ayurveda. I always say the scientific underpinnings or biological underpinnings, chemical underpinnings of Ayurveda. It will be just it's it's so applicable and so relevant that it will be really good if we can do this and convince people to use it for wellness. Yeah.
Absolutely, absolutely. The ashwagandha mode of action was really interesting, to understand with the research which is backed up.
Yeah.
Really wonderful. Yeah. So I have, one question. It's regarding Sroto Shodhan or Agni Deepan. So along with Brahmi or Ashwagandha, were there any other herbs given or anything to improve Agni or maybe diet
Yes. Yes.
or
Yes. Yes. Yeah. So this is a traditional Brahmi Krita recipe. As you
okay.
know, it also has other six herbs.
Hmm.
Trivrut, I think Trikatu. I'm not getting, but it has all other seven things. A little bit of Shankapushpi also, Shankapushpi also in it. So it's a Ashtanga Hridaya Brahmi Krita recipe which
Okay.
has which has other things also. In this case, when we do experiments with worms, c elegans, we only use the brahmi krita with brahmi swaras and brahmi kalka bakoponeri, because the all other herbs will get very sharp for the worms won't be able to tolerate it.
Okay.
So, because unlike us, the worms live in the plate, eat the same bacteria, it lives on that same bacteria as well. And we mix the food with the herbs with that bacteria, which is its food. So, those are tricks, experimental tricks we need to play. But for the clinical research, which I have shown you, we have used the traditional Ayurveda Brahmi Grita recipe, which
Oh,
also has other davyas. And in certain cases we have seen that panchakula churna or something which will be agni deepana will be good. And we what we do is that we request our participants to take brahmi gruta first thing in the morning, or ghee, whichever, plain ghee or
right.
brahmi, any formulation, first thing in the morning on empty stomach, and then with lukewarm water. So that we have kept constant. We don't change anupana. We keep anupana as lukewarm water only, because that again becomes a variable otherwise. So for this research, we have first thing in the morning on empty stomach brahmigata along with lukewarm water.
Okay.
So that's
And
it.
how long it was given? I think it was for a month,
Three
right?
months, 3 months,
3 months.
3 months.
3 months. Okay. Okay.
But but now, since people are seeing very good effects, people ask us whether we can continue for 6 months. But actually, Ayurveda says minimum 6 months. All our Ayurvedic colleagues said minimum 6 months. But we gave it for 3 months because uh, the compliance will reduce if you make the study longer and longer. Then
Mm.
uh, you will be knowing, having people retained in the clinical study itself is a challenge. Getting participants was a challenge, but now thankfully it is no more a challenge. There are many people who are interested by themselves and are coming to us. So that challenge is, we have somehow overcome that challenge. But otherwise, retaining the participant and getting both the data points before and after is a challenge.
Right, right, right. Absolutely. That's very true.
Yeah.
Yeah, and it was a very nice, interesting finding that definitely, supports Ayurvedic understanding or principles regarding the extract, like Brahmi extract, which is really helpful for younger adults because
Yes.
at that time there is more kapha, more pitta in the body, and Brahmi is going to help you to reduce that
Yeah,
pitta.
yeah, yeah. Yes,
and you
yes,
don't, there is not much vata aggravation. But as you
yes,
age, definitely there is more vata,
yes.
you need more ghee, like ghuta.
Yes. And then good thing is that Baku mind is internationally accepted. It's so that again, and always taking ghee for working person, sometimes it can get a challenge. So benefits of Brahmi extract can be definitely utilized. Like we can work on that more in the younger age.
Right, right, right. This is absolutely wonderful.
Thank
Great, great research.
thank you. So
Okay, so I would like to know if anybody has any questions. Okay, right. So thank you so much, everyone, for attending our webinar. Please watch and subscribe to our YouTube channel. So this recording will be uploaded to the YouTube channel. So definitely, you can watch it again and subscribe it so that you can also watch the earlier webinars and other presentations also.
yeah,
Please like, comment, and also share the videos with others who might be interested into knowing more about Ayurveda.
yeah,
Once again, I invite you to become a member and also fill out our volunteer form. You can also actually subscribe our Ayu update. So it's a newsletter that we publish. So you can also subscribe it to that and stay tuned with our projects as well as our events.
yeah. I would like to tell everybody that please do connect. We are definitely interested in collaborations. Even if we are many of you are in the US, you can we can always do something like online assessment. We can always do research together. So on various aspects, it may not be only this way of doing research. There can be many other ways of doing research, and we will be happy if we can collaborate. So I suppose that people who are practicing in Ayurveda in US, please do consider that how we please do think how we can work together. And Asavari, I have already told you that we can talk in detail and we can see how we can work together.
Absolutely. Wonderful. Thank you so much, Ashwini.
Yeah, my pleasure. Thanks
Welcome.
everybody for patient listening, and uh, let's see more in future. I any question? Okay. Bye bye.
Bye bye. Yeah. Thank you. Good night.